This is the third lesson for chapter 14. We're going to be looking at the last stage of how a gene is ultimately made into, or a gene is used to make a protein. And that process is called translation. Let's do a quick recap of where we left off though before we get into translation. Remember that we left off with the transcription of messenger RNA. And for a prokaryotic cell, this occurred in the cytoplasm, and for a eukaryotic cell, that messenger RNA was made in the nucleus but then exported out of the nucleus to ribosomes. So both prokaryotes and eukaryotes undergo translation at ribosomes. But there is a difference here because eukaryotes have different kinds of ribosomes. Some ribosomes are freely floating in the cytoplasm, and those ribosomes are going to make the proteins that stay within that cell. However, there's also ribosomes that are attached to the rough endoplasmic reticulum. Any proteins that are made on the rough endoplasmic reticulum ribosomes are going to be exported or leave that cell after they go through the other organelles involved in the endomembrane system. So definitely be sure you understand the difference between proteins that are made on the ribosomes that are freely floating in the cytoplasm versus the ones that are attached to the rough endoplasmic reticulum. But whatever the case and whichever the cell, the process of translation is similar. And so now we're going to look at that process in more detail. Similar to transcription, translation can be further broken down into 3 stages, and those are initiation, elongation, and termination. And so now we're going to take a closer look at those 3 stages, as well as the factors and molecules that facilitate each of those stages. Here is an overview of basically all of translation, including all of the different factors that are involved, such as the ribosome, as well as messenger RNA, transfer RNA, the resulting polypeptides made of amino acids. So now let's take a closer look at some of these new players that we haven't yet seen. So one of the new characters we haven't yet met is the transfer RNA. Transfer RNA is also a type of RNA like messenger RNA. So it is composed of nucleotides. There's a sugar phosphate backbone, and there are the nitrogenous bases. However, unlike DNA, where the nitrogenous bases are adenine, thymine, guanine, and cytosine, there's no thymine in RNA. Instead, there's uracil. So you might need to go back and review the base pairing rules from transcription to remember that RNA doesn't have thymine, but instead uracil. There's a note about that. And we're going to have a different base pairing rule actually in this lesson regarding translation between messenger RNA and tRNA. But first, let's just get oriented with tRNA. tRNA is a single strand, but notice how it's kind of coiled and wrapped upon itself, and there's hydrogen bonds forming between complementary bases, and that's what gives it its 3-dimensional structure, the interactions among itself. It also has a 3' and a 5' end, and like we've talked about this before several times, things can only get attached to the 3' end because that's where the hydroxyl group is, and that's where dehydration synthesis can occur. So on the 3' end is the amino acid attachment site. And the amino acid is not shown here, but soon you'll see where that, on this kind of cartoony version on the right, there's going to be a circle with an abbreviation for an amino acid attached there. The other notable structure of the tRNA is this part called the anticodon. Now, the anticodon is a sequence of 3 nitrogenous bases, and that's going to have a complementary codon for messenger RNA. And we'll see that later as we talk about the process of translation. Right now, we're just learning about these new characters that are involved in the process. Now, we looked at the ribosome in our first lesson. We saw that it's a very complex structure made of various proteins and RNA, ribosomal RNA. But now we're going to simplify it in terms of how the ribosome is involved in the docking of the messenger RNA, the attachment of the messenger RNA, because there's this messenger RNA binding site on the small subunit. And then on the large subunit, there's these other sites where the tRNA binds and where the amino acid that makes the polypeptide is going to be added to the growing polypeptide. So we do need to know these different docking sites. The P site in the middle is the site that holds the transfer RNA and carries the growing polypeptide chain. So I remember it as P for polypeptide. That's where the growing polypeptide will be. The A site holds the tRNA that carries the next amino acid to the growing chain. It's like the one that's in the holding pen waiting to be added. It's the arrival site. And then E is exit site. This is where the tRNA gets discharged or kicked off ribosome. Excuse me, I'll repeat that in case you heard that dinging. This is where the tRNA that already gave up its amino acid to the growing polypeptide gets kicked off of the ribosome once it reaches the E site. So now that we know the role, the structure and the roles of tRNA and the ribosome, and we already know about messenger RNA, let's see how they all come together through these 3 stages of translation, starting with initiation. How does it all begin? So it starts when messenger RNA binds to the small ribosomal subunit. And there's a special tRNA molecule, the initiator tRNA, that carries an amino acid called methionine. So this will always be the first tRNA that binds to the messenger RNA on the small ribosomal subunit at a particular location. Now, if we zoom in, you can see the complementary base pairing here between the messenger RNA in red and the tRNA in turquoise. So remember that the tRNA has what's called the anticodon that complements the messenger RNA codon. Okay, so I've just added a note to indicate that the tRNA UAC sequence of nitrogenous bases is the anticodon, and the codon of messenger RNA for that is AUG. This is what we call the start codon, as shown on the diagram at the bottom left. So with that, let me add a note about the base pairing rules between messenger RNA and transfer RNA for translation. So at the bottom of the slide, I just added a note, the translation base pairing rules. If messenger RNA has A, then the tRNA that complements that is U. If messenger RNA has U, then tRNA has A, and then cytosine and guanine are complements as well, as we've seen before, time and time again. That doesn't seem to change. All right, so think of that as the first step that needs to happen, the one shown on the left. Then once the small ribosomal subunit components are assembled, those 3 pieces are there, tRNA, messenger RNA, and small ribosomal subunit. Then the large ribosomal subunit attaches through a phosphorylation reaction. That's why the GTP, which you might recall from the 5' end of the messenger RNA, is used to help bind the messenger RNA to the ribosome. So that's where the energy comes from, through the phosphorylation reaction shown there. So now the ribosome is fully assembled. You can see that the tRNA, the initiator tRNA, is in the P site, which is where the polypeptide will continue to grow as more tRNAs come, bringing new amino acids. So now let's carry this on to the next step, elongation. Let's watch that polypeptide grow. So here you can see, although the word's cut off, codon recognition for the next tRNA based on whatever the messenger RNA codon is here. The tRNA with the corresponding anticodon is going to bind to the A site. Okay, and when it does that, notice that a peptide bond is going to form between the growing polypeptide, the amino acid that was most recently added, and the one here at the A site. So it actually shifts over to the tRNA at the A, but then that entire tRNA now carrying the growing polypeptide and the one in the P site all get moved to the left, shifts over. So now the growing polypeptide is in the P site, and the one that was in the P before that no longer has an amino acid shifts over to E, where it then translocates, which means basically it gets kicked off of the E site. So now the process can continue, and this continues until termination. When a certain stop codon is reached, and it's one of these three, either UAG, UAA, or UGA, and again, that's on the messenger RNA here in red, a certain release factor, which is basically a tRNA without an amino acid, will bind to the A site. Okay, now the growing polypeptide is still in P, and it will get pushed out along with the release factor, which then signals that the process is completed. The polypeptide is done, and now it will, if it's in the cytoplasm, it will be free, or it's off to the Golgi if this is part of the endomembrane system, if it was synthesized on the rough ER. The ribosomal subunits disassemble, and the messenger RNA and the release factor also disassociate from that complex. Now, if you recall, when in chapter 3 we talked about the different levels of protein structure, the thing we just looked at, that polypeptide, has primary structure. It's basically just the linear chain of amino acids. Further processing will likely happen, such as secondary structure, where it gets alpha helices, beta pleated sheets, because of the hydrogen bonds that form among the polypeptide with itself, or tertiary structure, additional bonds like ionic bonds or disulfide bridges, still again within that single polypeptide. But most proteins actually have quaternary structure, and so they're going to interact with other polypeptides to form a protein, a functional protein. So it's really not the end of the story after translation, because there's additional things that are going to be happening to that polypeptide.